My name is Maja Tarka, and I work here at the Department of Biology. I'm an evolutionary biologist, and I have a project on microevolution in tortoise beetles. So I would like to share some of our ongoing work. I wouldn't say that we are massively using computer vision yet, but we are starting to. So, tortoise beetles. Lots of variation and amazing diversity. But the type of variation that I want to talk to you about today is a bit less flashy, but super important, the one that is within species and within populations. So I'm interested in rate of evolution. So why do some populations evolve very fast? They can adapt over just a few generations, while other populations doesn't evolve and sometimes even go extinct. So in order to study evolution in populations, we need to know something about the variation, and there we need to know something about the phenotypic variation, but also about the genetic or the heritable variation. And the other thing we need to know something about is the selection that is acting on that variation. And when we have these two components, we can make some predictions of how fast evolution can proceed. So, in our project, we want to measure phenotypic variation in many traits. And the reason for measuring many traits is because traits are correlated, and these correlations can affect how fast evolution proceeds. So it can either facilitate or constrain evolution. We want to estimate natural and sexual selection. We want to estimate genetic variation, and then we want to predict the evolutionary responses. We want to do this within and between populations over time. So this is our green tortoise beetle roaming freely in its natural habitat. I don't know if you can see them. There's one over there, and three doing some hanky-panky up there. And so this is a common species in Europe and Sweden, and it is a specialist on mint, which is the nice thing because the mint creates some distinct habitat patches. And these beetles, they feed and they breed on their host plant, which means that we know where to look for them. And that also makes them quite sedentary, which means that we can track individuals throughout their lives. So that's that. And then another very good thing about them is that they have non-overlapping generations, which is convenient for evolutionary models. So we have closely monitored 3 populations this far in southern Sweden, and we go to each of these populations once or 1 to 2 times per week from May till September. And we've been able to track almost all individuals in these 3 populations. And in order to track them individually, we use some individual markings, so both on the dorsal and the ventral side. And at the end of each field season, we have done 2 this far. We had more than 90% individuals marked in all 3 populations. And when we find an individual in the field, we take it to our processing table or processing station, and once we have taken all the data that we want, we return it back to exactly the same position where we found it. So that's why we use these red flags. So we wanted to measure some morphological traits, and in order to do that, we take photographs of the beetles. And since we were talking about do-it-yourself tools, I brought our phenotyping tool. This is an old laboratory stand, repurposed. So we just fixate the camera here, so we have a distance that is the same all the time. And then we have taped some white paper, so we have contrast when we take the pictures. And this we can bring to the field, and it's much cheaper than the off-the-shelf solution. And then we have this Petri dish with a color reference and a size reference. And this is how, I lost one image. Anyhow, this is how the images can look like that we get from the field photography. So we take dorsal pictures of the beetles and ventral pictures of the beetles. So we actually just flip the Petri dish and take a picture through the plastic. And we have almost 23,000 images of 2,526 individual beetles. And the reason for taking this many pictures is because these are live animals, they don't sit still, and for being a beetle, I think they're quite flexible. They turn their head and do funny things with their body. So we want to try to standardize so that we can make proper measurements. And then to analyze all these pictures, to measure them, a master student, Erik Wendel, in our group, he has been the first one to try this out. And the type of morphology that we are interested in right now is just size and shape. So what Erik did was to annotate over 12,000 images, image files with beetle ID, and that was manual work, so I had to open each picture, see which beetle is in there, and note that down. He also made quality score of those images, so to find as beetles in a very good position, so to sort out the beetles that were not in a good position. And then he measured 1,274 individuals using Moritz nice phenopype software, and that has mainly been a lot of manual work, and that is something that we can later on use as a training data to make this process more automatized. So this is the type of work that Erik has been doing. So he asked the software to just find the beetle and then to make an outline of the beetle body. So this is only dorsal view pictures, and as you can see, it works quite well. Sometimes it works less well when the beetle has turned its head a bit. Then you get like an opening between the pronotum and the elytra. So these are the things that we need to fix. Often we have feet sticking out that needs to be corrected. Yeah. So that's that. And initially we were interested in 3 morphological traits, which is body length, area, and circularity. And circularity is the deviation from a full circle. So circle is 1 and 0 is a thin cigar. And this is the result, the only result that I will show you today. So the result of Erik's blood, sweat, and tears. So we have the 3 morphological measurements in 3 different populations. And as you can see, there is variation, and variation is good because then selection can act on this variation. So we can now start linking this to our estimates of survival. So for the future, we want to make the phenotyping more automated. So we need to train a model. I don't know which model or how to do that yet, but we will see. we also want to start using landmarks and segmentation so that we can get out other morphological features of the beetles. One that we are particularly interested in is the actual body size of the beetle, because if you flip it around, it looks very much like a turtle. So you have the beetle body there, and then we want to compare that to the actual shield size. And then I want to finish off with showing you two problems, and there might be a computer vision solution to this. I don't know, so I want to check with you. Maybe it's easier if we do this, continue doing this manually. But so one of the issues that we have is to do this annotation of the beetle IDs from the pictures. That's the first step. And we have started using QR codes, but the problem is that it's easy to do that when we find a beetle the first time. We have the label, they get their new ID, all is good. And then next time we retrap that beetle, suddenly we don't have that label anymore. So then we usually just do a handwritten note on which ID the beetle has, and we add that to the picture. And it can be, sometimes it's printed, a combination of letters and numbers, and sometimes it's handwritten. So my question to the experts is there a simple way to extract this from the pictures, or should we just continue doing it by hand? That's the first question. The second problem we have is a bit more complex, and so the annoying thing is that sometimes the marking doesn't stick, and when we retrap a beetle, it has lost some of its markings, which means that we need to go back to older pictures to try to see if this beetle was marked before, and compare that to all our so how that beetle looked like when it was marked the first time, right? And since this marking is done by hand, the marking end up in different positions and they have different shapes. So that's what we actually now use in order to identify the beetles with lost markings. And it works, but it takes a lot of time. So in my dream world, there would be, we would be able to take a picture of a beetle that has lost a marking, and then we just let it search all database of our pictures, and then spits out some pictures that fits the best. Yeah, so that would be a dream. The biggest dream would be if the markings wouldn't follow, but that's the case. So I would like to thank all people in our group, and all the people that have helped to collect all this data, and a special thank to Moritz, who has helped us to take the first stumbling steps in the computer vision world. And thank you all for listening.